Hello everyone and welcome to Brain Blitz Audios. Today we're here at another episode of SciBrief to look at yet another interesting scientific discovery. Today we'll be talking about the following news. Connexin proteins rally arteries to nourish the brain on demand. Now let's look at how the story goes. New research finds specific proteins that dilate arteries during enhanced brain activity. Let's read on. The human brain is a power-hungry machine, using about 1/5 of the body's resting energy. Unlike muscles or fat, it doesn't store much fuel. That means when neurons suddenly fire, such as the instant you recognize a familiar face in a crowd, energy must arrive immediately. To deliver this, nearby blood vessels widen and increase flow, a process called neurovascular coupling. But scientists were puzzled. Not just nearby vessels, but even distant arteries responded within fractions of a second, far faster than known chemical messengers could travel. Something else had to be carrying these urgent signals almost instantly. Researchers knew that neurons send signals to nearby capillaries, which in turn alert larger arteries upstream. This fuel delivery system seemed to require a messenger faster than neurotransmitters or hormones. The breakthrough came from Chenghua Gu's lab at Harvard University, which investigated the gap junctions connecting blood vessel cells. These are microscopic portals that let ions and small molecules pass directly from one cell to the next, forming a communication web along vessel walls. The team tested this by introducing serotonin into one cell and watching it slip rapidly into neighboring cells, revealing an extensive network, especially strong in arteries. The Harvard group pinpointed two proteins, CX37 and CX40, as being especially abundant in arterial walls. They suspected these connexins were responsible for the speed of the response. To prove it, they bred mice lacking these proteins in their artery walls. The results were striking. In normal mice, signals to widen arteries traveled over a millimeter in just a quarter of a second. In the modified mice, however, the same signal crawled at 1/3 of the speed. The slowdown became even more obvious when large regions of the brain became active, showing that gap junctions act like a scaling mechanism to coordinate fuel supply with bursts of neural activity. Other scientists weighed in on the findings. David Atwell, a neuroscientist at University College London, explained that this setup allows signals to travel along vessel walls and widen upstream arteries, effectively boosting blood flow before demand peaks. Brant Isaacson, a vascular physiologist at the University of Virginia, added that different vessels use different connexin proteins to pass along certain signals better, like having different pipes for different fluids. This flexibility means the vascular network can fine-tune responses based on what type of signal is needed. Together, their comments highlight the complexity and elegance of the brain's hidden plumbing system. The implications are wide-ranging. Anna DeVore, a neuroscientist at Boston University, noted that the study nailed down not only the mechanism but also the speed of vascular signaling. This knowledge is priceless, she said, for improving computer models that link brain activity to blood flow. Such models could enhance the accuracy of fMRI scans, which rely on blood flow changes to interpret brain activity. They could also help test new drugs in virtual simulations and guide therapies for vascular or neurological conditions. By pairing these insights with artificial intelligence, researchers could one day predict and perhaps correct blood flow mismatches in diseased brains. Finally, the findings here may shed light on health and diseases. Past work has shown mismatches between brain activity and blood flow, often described by imaging pioneer Amiram Grünwald as watering the entire garden for one thirsty flower. Delays in vessel widening can slow oxygen delivery, and this new study shows that gap junctions account for much of that lag, while slower chemical messages make up the rest. David Atwell pointed out that losing gap junctions with age or in small vessel diseases could reduce blood supply, although this remains unproven. Meanwhile, Isaacson suggested the work could open the door to drugs that activate connexins and to future studies on how the brain's 20-plus connexin proteins form mosaic networks to fine-tune signals. Ultimately, the research reminds us that the brain's lifeblood depends not just on electrical wiring, but on the quiet, rapid conversations of its vascular network. Now, that concludes today's scientific article. We hope you found this episode to be interesting and informative. This is definitely a new frontier in scientific exploration, particularly in the human body. If you want to learn more about such studies or about other interesting and informative content, please don't forget to subscribe to our channel, Brain Blitz Audios. Until the next webisode, take care, stay safe, ta-ta for now.